Greetings! This is Microbiology Chapter 1. It's an overview or an introduction to microbiology. So, microbes refer to a lot of different organisms that have to be seen with a microscope in order to be visualized. Um, so that's what microorganisms are. Germ refers to rapidly growing cell. It doesn't necessarily mean that it's going to be a pathological organism, but I think in our vernacular, germ does mean that. Now, the person who is responsible for the way that we uh, medically and scientifically refer to organisms like Staphylococcus aureus or Streptococcus pyogenes or Escherichia coli is due to a fellow named Linnaeus. And when we say Escherichia coli, the genus is always the first name, Escherichia, and the species is always the second name, coli. So whether you're talking about Bacillus anthracis or you're talking about Micrococcus pyogenes, then that first word, like with Streptococcus pyogenes, Streptococcus is the genus and Pyogenes is the species. And everything is in Latin. All medicine and science is Latin and Greek based. Oh, here we have Staphylococcus aureus. So in the case of the word, if we were very fluent in Latin, we would understand that staph is referring in Latin to the grouping, and the grouping is a grape-like cluster, and the coccus would refer to the actual arrangement of the round bacteria, or it would be these, the shape of the bacteria. So Staphylococcus would be a grape-like cluster of round cells, and then aureus, which is the species, refers to the golden color. And then we get to the major or a major chunk of microbiology, which is the study of bacteria. They are probably 10 to 100 times smaller than our own body cells. Hence, we have to look at them magnified 1,000 times. And bacteria are the first organisms that actually arose in the primordial sea 4 billion years ago. And we still have them, but they are a totally different kingdom than our own cells, and they are quite simple compared to our own much more complicated cells. So we call them prokaryotes. They actually don't have a nucleus, and the word karyot is a Greek word for nucleus, so this means before the nucleus. And the thing about bacteria is that they have a cell wall, they have a cell membrane, they have cytoplasm, they have things in this dissolved in the cytoplasm or in the cytoplasm. They have one circular piece of DNA. That's it. They don't have any organelles, they don't have any division of labor. And so the first cell was a quite simple cell, and we became so much more complicated when the prokaryotes became a new group called the eukaryotes with a nucleus. So the peptidoglycan cell wall is referring to a very rigid structural protective layer that lies outside of the plasma membrane. Division by bacteria are very simple. They basically clone themselves. On average, about every 20 minutes or so, bacteria are able to divide, and they merely divvy up their material, make a copy of their single circle of DNA, and then they just divide down the middle. And so that type of very simple division is called binary fission. Now, bacteria can use so many different types of high energy fuel, um, unlike ourselves. So we use organic chemicals, um, but they can also get energy from inorganic chemicals like hydrogen sulfide. If you ever smelled a rotted egg, that's hydrogen sulfide gas that you're smelling. And we certainly cannot take hydrogen sulfide gas and have it come into our bodies and make tons of energy and thrive on H2S gas, but bacteria can. Photosynthesis, some of the bacteria do have the ability to do photosynthesis just like plants. So as you can see, they're very versatile. All right, so viruses is another big group, but viruses are actually not considered living because they cannot divide on their own. They have to parasitize a cell and use a cell and the cell's machinery, the cell's energy, the cell's building blocks in order to kind of usurp them and build a new virus based on their viral DNA or RNA, which contains the instructions on how to do that. So that's what they mean by acellular. They're either a DNA or an RNA virus. They have a protective outer coat, like a core. You might have a second coat, like a lipid envelope right here. And all of those coats or cores or capsids or whatever you want to call them, they're just to protect that nucleic acid that contains the instructions that lie in the middle. And so, right, they can only replicate themselves. Now, they are actually smaller than bacteria, so they are 10 to 100 times smaller than bacteria. So you really need an electron microscope to see them. And they are only replicated when they're in a living host cell. Now, everybody else is eukaryotic. So you're going to find all kinds of other types of microbial parasites, things like fungi, yeast, protozoal parasites, and they are eukaryotic cells. They are with a nucleus. And that was a big advance. And that also means that it has organelles, it has division of labor, it is very complicated. It is able to do things in a much more sophisticated manner than bacteria. It also makes it difficult to kill. But some of them are multicellular, some of them are unicellular. And things like flatworms, again, yeast molds, brown worms, and other types of parasites that very often are oral fecal in origin, and that's how they spread. And they are microscopic stages, and they have complicated life cycles. Now, the most early life on Earth were bacteria, but they actually weren't seen until the microscope, very primitive microscope that was discovered by Leeuwenhoek in 1673. They're just far too small for us to see without assist of something like a microscope. So Antony van Leeuwenhoek, don't memorize the dates. This is his actual microscope. And he was just in love with looking at pond water or teeth scrapings and to see his little, what he called animalcules, tiny little animals that were in the water. And now he realized that they were probably amoeba, paramecium, probably very, very tiny protists. Now, believe it or not, back in about the 1850s, there was a huge debate about, did these organisms spring to life on their own, that's spontaneous generation, or were they actually coming from other, did they have to come from other cells? So was it biogenesis, life from life, or was it spontaneous generation, life from death? And Louis Pasteur finally laid the argument to rest, and the bacteria, although you can't see them, they are carried on dust. So that when you are in in surgery. You create a sterile field, and that field has to be a dust-free environment because that is how bacteria are able to invade, and that's how they are able to be carried. So when Louis Pasteur had a flask, and one flask was opened and one flask was sealed, and he heated them to boiling, and only the flask that was open became contaminated with bacteria because the dust fell from, the room into the broth, and then it was carrying bacteria, and the bacteria grew. But if he sealed off the flask, dust couldn't get in. And since the nutrient broth had been boiled so that nothing was living in it, it did not get contaminated and remained sterile. So the theory of biogenesis is what we continue to use today. Then, and again, you notice that you're in like the 1850s to the 1900s, and a lot was discovered in microbiology at that time. Joseph Lister was famous for using chemical disinfectants before surgery. Robert Koch became famous for proving that microbes could actually cause a disease. And he was able to make what are called Koch's postulates. And it's a four-step process by which you are able to prove that bacteria are the cause of a disease. Excuse me. And Edward Jenner is the father of vaccination, and we'll talk more about that when we get to the immune system. And finally, chemotherapy. Between World War I and World War II, the British government was able to pour a lot of money on Alexander Fleming's observation. Now we're in the 1928s. And what he observed was a contaminated plate. It was contaminated with a mold called Penicillium notatum, right here around 12 o'clock. And he came back and he saw what other people saw, but he made that intuitive leap. And he saw the zone of inhibition. So there is a zone where there's absolutely no bacteria growing, and then the bacteria are growing away from that. And he correctly surmised that the mold was making a chemical that diffused in the agar that could inhibit bacteria. And that was the discovery of penicillin. And since in World War II more soldiers died of bayonet infections than actually from bullets, that the government kind of knew that there was another world war on the corner, and that whatever country could indeed develop chemicals like antibiotics, then they would win the war. And probably after World War II, I would say maybe 1945 to 1950, penicillin became one of the most prescribed drugs in the world. And that's it. So I will see you for the next chapter.